I'm Suzuki from Tokyo University. Today, I'm talking about a novel drug delivery system for cancer therapy. Recently, some liposomal drugs have been approved for cancer therapy. However, liposomal drugs are limited to deliver into tumor tissue. So, it is necessary to develop a novel drug delivery system. In this study, we tried to develop doxorubicin encapsulated liposome-loaded microbubble to solve the drug delivery issue. In this presentation, we call doxorubicin encapsulated liposome D-Lipo and D-Lipo-loaded microbubble D-Lipo-MB. When D-Lipo-MB in blood flow are exposed to ultrasound, the D-Lipo would be released from microbubble due to crops of them. Thereby, the D-Lipo could be delivered into tumor tissue. In this study, we assessed the feasibility of active drug delivery by the combination of D-Lipo-MB and ultrasound. This slide shows the preparation method of D-Lipo-MB. This lipid suspension and perfluorobutane gas were put in the vial. Then the vial was shaken with vial mix to form butynylated microbubble. This microbubble was mixed with avidin to make the avidin-modified microbubble. Finally, this avidin-modified microbubble was incubated with butynylated D-Lipo to prepare D-Lipo-MB. This shows the fluorescence image of D-Lipo-MB. We could observe that D-Lipo was loaded on microbubble. Next, we assessed the intracellular delivery of doxorubicin with D-Lipo-MB and ultrasound. D-Lipo-MB was added to pancreatic cancer cells, PAN02, and then ultrasound was exposed. After 4 hours, intracellular doxorubicin was observed with confocal laser scanning microscope. This is results. In the combination of D-Lipo-MB and ultrasound, the doxorubicin was observed in the cytoplasm. This result suggested that the combination of D-Lipo-MB and ultrasound could promote the doxorubicin delivery into the cells. In the next step, we assessed the growth suppression of cancer cells. D-Lipo-MB was added to PAN02 and ultrasound was exposed. After 1 day, the cell viability was assessed. In this experiment, we also examined empty lipo-loaded microbubble. We call this Lipo-MB. This is result. In the treatment of D-Lipo-MB or Lipo-MB without ultrasound, the cell viability didn't decrease. On the other hand, in the D-Lipo-MB and ultrasound combination group, the cell viability significantly decreased. This result suggested that D-Lipo was released from microbubble by ultrasound exposure and effectively delivered. This is conclusion. We succeeded to develop D-Lipo-MB that had D-Lipo on the surface of microbubble. The combination of D-Lipo-MB and ultrasound promoted the delivery of doxorubicin into the cells. And the combination of D-Lipo-MB and ultrasound could suppress this cancer cell growth. Therefore, this combination would be an effective drug delivery system for cancer therapy. Thank you for your attention.